This is the second part of our introductory lecture. In this video, we'll go through the mechanisms of cell death, of which there are really two types. We have either uncontrolled cell death, which we call necrosis, and we have controlled cell death, which is apoptosis. Necrosis is caused by injury, most commonly. So this is considered a pathological form of death. So let's say you get a cut on your hand here. That cut causes necrotic death of the skin cells. What's going on there is that we rupture the plasma membrane. So whatever caused the cut physically slices through the plasma membrane, causing the cell to explode and leak out its insides. When those insides leak out, that triggers an inflammatory response. And what you'll notice is that the areas around your injury swell and turn a bit red. That's inflammation. Inflammation can be a very bad thing, especially in the brain where we have, again, a set volume. It's encased in the skull, so if we start swelling in the brain, that's going to increase pressure. And we'll see throughout this class that doesn't end well. Necrosis can also occur without having something slice through the membrane. In some cases, the cell simply swells up so much that it, like an overinflated balloon, pops, causing the plasma membrane to rupture. So whether it's an external physical force or an internal physical force, necrosis happens at the membrane. Now, apoptosis, on the other hand, is a form of controlled cell death. And there's a couple of reasons we might want to remove cells. If they're highly damaged, for example, we run the risk of forming cancer cells, which will be a big problem for the rest of the organism. So it's best to have any highly mutated cell go ahead and kill itself in a controlled manner. There might also be some cells that we don't want. Hey, where did that hand come from in the first place? Well, it developed from an embryonic structure that was an awful lot like a fin. And that's great if you're going to be a fish. Now, we don't develop fins, we instead develop hands. And the way that we do that is by sculpting away the cells that we don't want through apoptosis. So we'll just kill off the cells that shouldn't be there, and the next thing you know, you've got yourself, well, we'll call it a hand. You get the point here. Sometimes we want to remove cells. And truth be told, a lot of your neurons that you're born with don't make it to adulthood. That's a part of maturation, is killing off unwanted neurons. Now, when we kill off cells through apoptosis, what we want to do is break it down into little tiny digestible fragments, and there are enzymes called caspases that will carry this out for us. Now, the site of initiation, rather than being the plasma membrane, is generally the mitochondria. When the mitochondrial membrane is permeabilized, this activates caspases. The cell is then reduced in size, broken into little tiny fragments, including the nucleus. And these don't trigger an inflammatory response. Instead, the plasma membrane is just simply flipped inside out on all these little fragments so that the nearby macrophages just detect it as a little tasty bit, not some foreign invader or a sign of damage. So there's no inflammatory response. That is, we can kill off the unwanted cells without disturbing our neighbors. If you prefer a visual representation, here it is. Necrosis again on the left, apoptosis again on the right, to match up with the table. As far as necrosis goes, two options. We can have traumatic injury. That's pretty straightforward. Some object ruptures the plasma membrane. We can also have death from within. We'll explain that a little bit further here. We call this oncosis, that is osmotic necrosis. So the problem here is water. Too much water enters the cell, and that's in neurons, typically caused by excessive ion currents. So if we move a bunch of sodium, let's say, into the cell, so let's get us a cell here. If we start to fill this thing up with sodium, what that essentially does is decrease the effective concentration of water inside the cell. This then creates an osmotic gradient, so water comes in. That causes the cell to swell and swell until it eventually pops. Let's get some of this junk out of the way. All right, here we're going to see oncosis occurring in these cells. Look at them swelling up. They're getting much larger, and then pop, pop, pop. So those three are done for, and then their neighbor follows suit. So those cells are done. Apoptosis is going on over here. Notice this cell's breaking up into little tiny fragments. This is a more active process because the cell is carrying out the apoptosis over here, whereas over here, necrosis is a bit more passive. Because more ions came in, water simply followed by diffusion, causing the cell to swell and rupture. Couple reasons why we might have excessive ion currents. First of all, we don't have enough ATP. So normally when a cell brings in some positive charge, it'll then pump it back out against its concentration gradient, but this costs ATP. If we don't have ATP, we don't remove the ions, so they build up. The other option is that we have an excessive release of glutamate, that excitatory neurotransmitter. Glutamate is the neurotransmitter that opens up ion channels, bringing in the positive charge. So whether it's excessive influx of ions or insufficient removal of ions, either way, the cell will fill with water and pop. Now, there is no pop in apoptosis, other than when you say it. Instead, this is death from within. It can be stimulated by external factors, but we won't really consider that. Know that it happens, but forget about it for the exam. Apoptosis is generally stimulated within the cell, and the site where all the action is going to take place is the mitochondria. So here we have a single mitochondrion here. Notice there's two membranes. In the intermembrane space, we've got a few nifty things. The one that we care about here is cytochrome C. So I've just color-coded this for your ease. The red circles are cytochrome C. Now this is a part of the electron transport chain. So the whole point of mitochondria is to generate a bunch of ATP. And cytochrome C is one of those components of the electron transport chain, but it's not embedded in the membrane. It's soluble. It can float around in that intermembrane space. It helps transfer electrons through the electron transport chain. Now, the mitochondrial membrane changes its permeability based on a couple of things. First of all, if the cell starts to fill up with calcium, this will get buffered by the mitochondria. Let's say we have a whole bunch of calcium within the neuron. Well, the mitochondria is going to create something called a mitochondrial transition pore. MTP, mitochondrial transition pore. And that will allow cytochrome C to leak out. Another regulator of mitochondrial membrane permeability are the BCL2 proteins. Remember, if something happens in life, a protein did it. BCL2 proteins come in two general flavors. Those that kill us, that is the pro-apoptotic BCL2 proteins, those permeabilize the mitochondrial membrane. And then there are the anti-apoptotic BCL2 proteins, and those stabilize the mitochondrial membrane. They prevent permeabilization. Okay, so let's consider two scenarios. On the left, we have an excess of pro-apoptotic BCL2 proteins compared to anti-apoptotic. what the pro-apoptotic BCL2 proteins is insert into the membrane, making a pore, just like what happens if we have excessive calcium. Cytochrome C leaks out of this pore made by the pro-apoptotic BCL2 proteins and forms the apoptosome, along with another protein, APAF1. The apoptosome is the structure that stimulates caspases, the enzymes that carry out apoptosis. So if we either have prolonged elevations in calcium or we make a whole lot more pro-apoptotic BCL2 proteins than anti-apoptotic, we get apoptosis. Let's consider this scenario on the right. If we have an excess of anti-apoptotic BCL2 proteins, that's the green jelly beans here, they bind to the pro-apoptotic BCL2 proteins and prevent them from inserting into the membrane. That is, the mitochondrial membrane stays intact. Cytochrome C up here can't leak out. There's a membrane in the way. So we don't form the apoptosome, we don't activate caspases, and we do survive. Now that balance of pro versus anti-apoptotic BCL2 proteins is regulated by neurotrophins. And this is a group of proteins that are there to keep neurons alive. And they bind to two broad classes of receptors, which we will oversimplify a bit and say that TRAC receptors are anti-apoptotic and P75 neurotrophin receptors are pro-apoptotic. And this is true except when it's not. We'll run with this generalization though. So my neurotrophins are up here. Those are the purple jelly beans. If we have a moderate amount of neurotrophins, those activate TRAC receptors, and TRAC receptors will indirectly change gene expression. So they'll eventually activate transcription factors that turn on anti-apoptotic BCL2 proteins. That's those green jelly beans from the last slide. If we stimulate P75 receptors, generally we think of this as happening because of an excess of neurotrophins. Then P75 stimulates the expression of pro-apoptotic BCL2 proteins. So we have to get the right concentration. Not too much. So if we have a very high concentration of neurotrophins, we start to turn on P75. That'll kill you. If we don't have enough neurotrophin around, well, then we don't stimulate either receptor, and we take the default pathway, which is death. Neurons only survive if there's a moderate level of neurotrophin signaling, so that we have preferential activation of TRAC receptors. Let's preferentially activate them now. Look at those activity lines. This is a happy neuron. Only in that case do we stimulate TRAC receptors and keep a high level of anti-apoptotic BCL2 proteins around, keeping our mitochondria intact. Apoptosis is a pretty quick process. I'll get this out of the way. What we're looking at here are some cells that aren't long for this world. The red is showing us the mitochondria. The green is going to show us active caspases. Now you'll notice there's kind of a green background, but the cell isn't that green yet. Wait for it. And there you go. Now it's dead. So pretty quickly we go from living cell to dead cell. And notice it's breaking off these little tiny blobs. Those are going to be digested by nearby macrophages. No inflammation required. Now what leads us down the road to necrosis or apoptosis? Well, that's what the rest of this class is about. A very common cause of cell death in neurons is glutamate toxicity. And this can cause either necrosis or apoptosis. Inflammation is also a very common way of killing neurons. If we have swelling, the mechanical pressure can be enough to stimulate necrosis. Alternatively, inflammation can trigger glutamate toxicity, leading to apoptosis that way. The remaining three mechanisms that we'll see pretty commonly don't have much to do with necrosis. Instead, they're going to trigger apoptosis through a couple of different ways. If we don't have enough neurotrophic support, well, then we're not going to have enough anti-apoptotic BCL2 protein expression. That'll cause apoptosis. If our mitochondria don't work, well, they're either permeable, leading to apoptosis directly, or we don't make enough ATP, and that'll kill you as well. Protein aggregation disorders impair our ability to make proteins and trigger what we call the unfolded protein response. This is a long way of saying apoptosis. It's the same thing. So if a cell can't fold its proteins properly, it can't do anything properly because proteins do everything in life. And therefore, the logic built into a cell is if I see a lot of unfolded proteins, my response should be apoptosis. So we'll walk through each of these mechanisms, seeing the cause and seeing the consequence. We'll get this all throughout the class, but it's a great idea to go ahead and get familiar with these mechanisms now. So we'll start off with glutamate toxicity. Couple of things that can cause it. Obviously, a high level of glutamate will cause it. That's the problem. But for glutamate toxicity to occur, we have to activate NMDA receptors. All right, quick bit of terminology here. There are two broad classes of glutamate receptors, NMDA and non-NMDA, which we will call AMPA. Now you'll notice that these IV plots look quite different between the AMPA receptor and the NMDA receptor. The AMPA receptor has a nice straight line. I get a current, that's on the y-axis. I is current, V is voltage, that is membrane potential. So let's say I'm over here at rest. I open up an AMPA receptor, I get an inward current. That is, ions rush into the cell, depolarizing me. And that's why glutamate receptors are excitatory. Okay, same thing should happen over here, but no, we get no current through NMDA receptors at rest. And that's because they have a magnesium block in the way. All right, let's imagine an NMDA receptor. So just a couple of jelly beans that'll come together and form a pore in the membrane. At rest, the cell is negative, and that's going to attract that positively charged magnesium into the cell. It tries to get in through NMDA receptors, but it can't quite fit. Kind of like Winnie the Pooh trying to crawl through the hole in the tree trunk, it gets stuck. So the magnesium ion sits in the ion pore, preventing any ion movement until we can repel it. If we start to depolarize the neuron, that is, we bring in some positive charge, like repels like, and we push that magnesium ion out of the way, let's get it out of the way, opening up the NMDA receptor and allowing ions to flow through. And that's why we see this J-shaped curve here. Hey, over here where we're pretty well depolarized, it's a nice straight line. That's great. But down here in those negative membrane potentials, notice we lose our current. It doesn't just keep on going like the AMPA receptor does. Instead, we start to attract magnesium in, and that blocks it. Now, why is the NMDA receptor important? Let's take a look down here. The y-axis is showing us the open probability. That is, how long the receptor is open, because X is showing us time. So once we apply some glutamate, the AMPA receptor opens up very quickly and then almost immediately desensitizes. So if you look at the area under the curve, AMPA receptors don't move a whole lot of ions. NMDA receptors are a completely different story. Yeah, they're a little slower to get up to max, but they stay open much longer. So the area under the curve here for the NMDA receptor is much greater. That is, NMDA receptors just simply move a whole lot more ions. That makes them able to kill us. Either we get that oncosis, or included in those ions is calcium. And as we saw, that can cause the mitochondria to permeabilize. So depolarization is therefore necessary for glutamate toxicity, because we have to allow for NMDA receptors to be activated. Okay, so what can cause that depolarization? Well, many things. So surprisingly, if you don't have enough ATP to pay the bill, you depolarize. You might think that a neuron that doesn't have enough ATP would just stop working. And it does, but only after it works itself to death, pretty much. So let's look up here at these data. The x-axis is showing us time in seconds, and the y-axis is showing us the membrane potential. You'll notice this neuron is fairly active. That's what all the spikes are here. Those are action potentials. After about 45 seconds, they apply 1 micromolar of wabane, which is a sodium-potassium pump inhibitor. This is mimicking running out of ATP. If we don't have enough ATP, we can't fuel the pumps. If we don't fuel the pumps, well, then the positive charge that we bring in lingers around. We'll think of it that way for now. I'll explain it a bit differently and more truthfully in neurobiology. But just think that the positive charge hangs around. That's what's going on here. We're accumulating positive charge, and look where the membrane rests. It starts to creep up and up and up and up until that neuron is just chronically depolarized. No more action potentials. Don't worry about why. Just realize that this neuron is highly depolarized. So if it gets a dose of glutamate over here, we're going to see a whole lot of NMDA receptor activation. So my NMDA receptors are now able to be active. That can move a lot of ions, and that'll kill you. That's not all. If we fail to clean up potassium, let's say we have some reactive gliosis, the excess of potassium depolarizes neurons. All right, we're looking at something similar down here. Instead of looking at time on the x-axis, now I've just plotted the concentration of potassium outside the cell. This is about the physiological level, and so that's our normal resting potential. You'll notice that resting membrane potential creeps up. It depolarizes as we increase potassium concentrations. And potassium concentrations in excess of 10 millimolar certainly occur in pathological states. So this isn't just something that will happen in the lab. This occurs in pathological states. Those depolarized neurons, same story as up here. Now we're removing that magnesium block, and now my NMDA receptors can be active, leading to glutamate toxicity. Of course, you can't have glutamate toxicity without a whole bunch of glutamate, and that can come from a variety of sources. Hey, those depolarized neurons that are firing a whole bunch of action potentials, they're spitting out glutamate probably. Anytime we activate microglia, they can spit out glutamate. If we have a bunch of inflammation and T cells enter the brain, they spit out glutamate too, and believe it or not, tumor cells spit out glutamate. So we have several sources of glutamate. Another possibility is that reactive astrocytes don't pick up glutamate as much. So when the neuron spits glutamate out into the synapse, normally it's cleared by astrocytes. When they become reactive, we stop picking it up. What that does is increase the amount of glutamate, leading to, you guessed it, glutamate toxicity. Okay, so what's the problem? Well, could be oncosis. Here we've got our nice little neuron. Ions are color-coded red. I made the water blue because water's blue in my mind. Okay, let's stimulate some ion currents. What happens? The red ions flow in. Hey, water's got to follow. It's just osmosis. Continue this. Eventually, just like if you blow a balloon up too much, the membrane ruptures. We can only stretch it so far. And then you've got yourself necrosis, leading to inflammation and causing problems for your neighbors. So don't do that. The other option is apoptosis. Okay, maybe you're keeping up with the ions. You're pumping them out. That ain't free. That cost ATP. You're burning through your ATP, and now you got to make more. Here's the deal. Every time you make ATP, you run the risk of producing free radicals. These are also called reactive oxygen species, or ROS. Same thing. Free radicals have unpaired electrons. This is unstable, so they donate the electrons willy-nilly onto whatever part of the cell is nearby, whether it be membrane, protein, carbohydrate, DNA, doesn't matter. Free radicals damage the cell. This is clearly not a good thing. The other thing that glutamate can do is elevate intracellular calcium levels, creating that mitochondrial transition pore. Cytochrome C leaks out. Here's someone else's cartoon of this. It dimerizes with APAF1 there, forming the apoptosome. We turn on caspases and apoptosis. The other thing that can happen is if we have too much glutamate floating around, it can diffuse away, right? Or if we fail to clean up the glutamate, it can diffuse away. So if we're normally releasing our glutamate right here, that glutamate is primarily going to stimulate the NMDA receptors nearby. If we stop cleaning it up, though, it can diffuse away and stimulate extrasynaptic receptors. These are essentially error detectors that neurons have. If we detect glutamate outside of the synapse, there's a problem with that synapse. What we should do is eliminate it, because clearly it's not functioning properly. So let's get rid of the synapse. If you stimulate a bunch of extrasynaptic NMDA receptors, then the logic of the cell is something is wrong with me. I need to get rid of myself. So those extrasynaptic NMDA receptors are going to change gene expression. So now we're down in the nucleus down here. We'll turn on some transcription factors that'll modify the expression of clearly some very bad genes. Hey, BIM, what the heck is that? That is one of those, let's make it yellow. One of those pro-apoptotic BCL2 proteins. That's clearly going to end poorly. Inflammation is another common issue that we'll find in this class. The cause ordinarily is either infection or damage. That's it. Now remember, we have a resident immune cell, the microglia, floating around in our brains. Well, floating, right? It's living in our brains. This is able to detect infection or damage. More details on this later. And when it does, it triggers an immune response. So it's going to spit out pro-inflammatory cytokines. This will trigger gliosis. It can also then lead to inflammation, modifying blood flow, etc. Besides infection and damage, tumor cells can stimulate inflammation. One of those pro-inflammatory cytokines is TNF-alpha, tumor necrosis factor alpha. And it's called that because they found a whole lot of it floating around tumors that were surrounded by necrotic tissue. So that's why it's called tumor necrosis factor. Alrighty, so early on, tumor cells might stimulate inflammation indirectly by turning on immune cells that identify it as a problem. But later on, more advanced tumors directly release pro-inflammatory cytokines like TNF-alpha. And that's what we're looking at here. So here we're looking at some ovarian extracts, normal ovaries. We got 10 of them here, and then 10 ovaries that had tumors. And the darkness of each of these dots, this is called a dot blot assay, shows us about how much TNF-alpha was around. Over here, this is just a loading control, ubiquitin. You can kind of forget about it. It's just to make sure they actually loaded about the same amount. And what we can see here, there's a whole lot more TNF-alpha. If you prefer bar graphs, here you go. There's that tiny, sexy p-value right there, clearly less than 0.05. So there's a lot more TNF-alpha spit out from these tumors. TNF-alpha then triggers inflammation. Hey, what's so bad about inflammation? Well, inflammation leads to reactive gliosis. Right? Don't forget that whole feedback loop between microglia and astrocytes. So once we have inflammation, we then create reactive glia. That includes microglia and astrocytes. Microglia, when they're reactive, they spit out glutamate, leading to glutamate toxicity. They also create a bunch of free radicals, spitting those out, which can damage nearby neurons. Astrocytes stop picking up potassium and glutamate, and heck, they can even spit out glutamate, again leading to glutamate toxicity. See the last couple of slides. The other issue with inflammation is that it affects the vasculature. Sure, it sucks to spit out glutamate and stimulate apoptosis in neurons or oligodendrocytes, but that's not it. That's not the only problem. If we permeabilize our blood-brain barrier, then things can enter the brain that shouldn't. White blood cells, for example, which spit out glutamate, and by golly, we're back to glutamate toxicity again. Another problem, just fluid. Fluid can come into the brain, increasing intracranial pressure and potentially causing ischemia, making us run out of ATP, causing excessive depolarization of neurons and leading to either oncosis or apoptosis through glutamate toxicity yet again. And here we can just see the permeabilization of the blood-brain barrier. Normally, white blood cells shouldn't be able to leave, but hey, we've got some reactive astrocytes, and so now they're not maintaining the blood-brain barrier. Alrighty, what about those neurotrophic factors, right? You need the right amount, not too much, not too little. So what if we have too much? Well, one thing that can cause too much is hyperactive neurons, because here's the deal. Neurons release neurotrophic factors to keep their neighbors alive, and they do it in an activity-dependent manner. Here's the logic. If a neuron is in a network, that is, its activity is stimulated by another neuron, its formed functional synapses keep it alive. However, if a neuron is never active, why are we keeping it around? Kill it off. The neurotrophic support allows us to do that. So we want to have enough activity, not too much. With a moderate level of activity, we release a moderate amount of neurotrophic factors and stimulate those TRK receptors. On the other hand, if we're hyperactive, we spit out a lot more neurotrophic factors, which biases us a bit more toward P75. Another thing that can impair neurotrophic support is anything that interferes with our ability to make proteins and move them around in the cell. So protein aggregation disorders, or anything that impairs gene expression, mutations, for example, in transcription factors, those can change which genes we make and how much of them we make. If we don't read the DNA correctly and make the correct amount of mRNA, we won't make the correct amount of protein. Protein aggregation disorders interfere with the folding of those proteins. Structure is function. If we don't fold the protein up correctly, it doesn't do its job. And remember, proteins do everything in life. Neurotrophins are proteins. Their receptors are proteins. So anything that interferes with protein production can interfere with neurotrophic support. And an important consideration for neurons is moving those neurotrophin receptors around. Okay, if you're a motor neuron, how do we know if you should survive? If you form a synapse with a muscle, you can survive. If you don't, you're not much of a motor neuron, you should die. So the logic here is neurons that synapse with muscles get neurotrophic support and survive. But here's the deal. Those neurotrophin receptors are really far away from the nucleus. That's where axonal transport comes in. So recall this from the last video. We got the red dots. Those are TRK receptors running on down the axon, and red dots are also coming back. So new TRK receptors are transported with anterograde, and the active TRK receptors are brought back to the nucleus to keep the neuron alive by retrograde axonal transport. So anything that damages microtubules that is the roads that motor proteins use for axonal transport, or the motor proteins, or the attachment of cargo to motor proteins is going to interfere with axonal transport and thus interfere with neurotrophic support. Here's some data showing us the effects of alpha-synuclein. This is a protein that is implicated in Parkinson's disease. So we're looking at the effects of alpha-synuclein on retrograde axonal transport. The y-axis is showing us the velocity, the average velocity of these little dots. So they went and they counted the dots, saw how fast are they moving. Here's the typical values for normal mice. So in TG, that means not transgenic. TG are transgenic mice that have a whole bunch of alpha-synuclein. So it's a model for Parkinson's disease. We can think of it that way. And notice we're moving those neurotrophin receptors back much slower, about half the speed. And that's because a lot of them are stalled. Hey, if we don't move our neurotrophin receptors back to the nucleus, we don't continue to express anti-apoptotic BCL2 proteins, and therefore we go down the default of death. The consequences are pretty darn straightforward. If we have enough neurotrophic support, we stay alive by creating anti-apoptotic BCL2 proteins. If we have too much or too little, we either don't make the anti-apoptotic BCL2 proteins, or we make an excess of pro-apoptotic BCL2 proteins. Either way, that cell is toast. Now, of course, the mitochondria can themselves be the problem in these disorders that we go through in this class. So if the mitochondria doesn't work, cell doesn't work. In ischemia, the big problem here is that we stop blood flow to some part of the brain. This deprives it of glucose and oxygen. Therefore, the mitochondria don't work because we need oxygen to accept the electrons from the electron transport chain, and we need the glucose to make the electron carriers that fuel the electron transport chain. So we run out of ATP, and we're back to glutamate toxicity. Now, without that oxygen around, right, we've gone through ischemia, then the electrons don't get put into water. Instead, those free electrons float away and form free radicals, or the reactive oxygen species, like superoxides and things like that. These then go and damage the cell willy-nilly. Mitochondrial dysfunction can also occur because the cell just simply doesn't clean up the mess. Now look, everything runs out in life. Everything wears out. This is true for mitochondria as well. And so we're always sniffing around to see, do I have any damaged mitochondria? If so, get rid of them, because a damaged mitochondrion will kill you. Cytochrome C leaks out, the apoptosome forms. It doesn't end well. Now, there's a couple of proteins involved in quality control monitoring here. And so pink 1 is our first line. It's color-coded pink over here. Very cute. Pink 1 is normally always slamming into the mitochondria, burying into the outer membrane, and then always being removed. It's always removed in healthy mitochondria. In unhealthy mitochondria that are unable to clear pink 1, it accumulates. Pink 1 creates the binding site for the second protein, parkin. That's the blue one here. So first we coat it in pink 1, then parkin binds, and what parkin does is add a whole bunch of these ubiquitin tags onto the mitochondria, and that is a sign for the cell to destroy it. So things that get ubiquitinated are tagged for destruction. So we then load that mitochondrion into a lysosome and break it down before it gets leaky and kills the cell. Mutations in pink 1 and parkin allow those damaged mitochondria to accumulate, and these are associated with an increased risk of Parkinson's disease. So we'll revisit these later. So we need to be able to identify and then break down damaged mitochondria. Maybe we have a problem identifying them because of impaired quality control. And heck, maybe we have a problem degrading them. In protein aggregation disorders, the proteasome, which is a part of this degradation system, doesn't work properly anymore. So sure, you can ubiquitinate your mitochondria all you want, but if you don't break them down, they're still going to leak, and it's still going to kill you. Either we have leaky mitochondria that don't make enough ATP, leading to chronic depolarization, that then causes excitotoxicity, that is glutamate toxicity. The other option is, well, if our mitochondria just aren't working well enough, aren't working fast enough, let's say because of ischemia, then we're going to get oxidative damage. We make all those free radicals, and then they non-specifically damage the cell. And if you damage the cell enough, that triggers apoptosis. Protein aggregation disorders will come up in this class as well. And these are caused either by the formation of sticky proteins, that is, cells that have either a bunch of beta sheets. Don't worry about that. Think of this as Velcro, and the cells and the proteins stick together. Polyglutamine is really common for creating these sticky motifs. Or the protein can be sticky because it has a lot of hydrophobic amino acids. This is the case for A beta. If the peptide is hydrophobic, it tends to fall out of solution and aggregate because it's not as water soluble. Another possible cause of protein aggregation disorders is just simply misfolded proteins. Prions are a great example that we'll see later on in this class. So it could be a problem with the protein, the prion, or it could be that proteostasis network. So when we make a polypeptide, that's not the end of the story. We have to fold that polypeptide correctly into whatever shape gives us the function that we want. There are proteins called chaperones that assist us from going to this disorganized noodle into the properly folded protein. Chaperones are there. If we have mutations in chaperones, we're less likely to properly fold, leading to an unfolded protein response. Now, of course, if we have mutations in the proteasome, which is normally there to degrade damaged or misfolded proteins, well, then they accumulate, leading to the unfolded protein response. Now, those, if we have sticky proteins, one of the things that they can do to kill us is to screw up that proteostasis network. So here we can see polyglutamine, that's the poly Q here. Those are the green dots. Green is showing us polyglutamine, and red is showing us components of the proteasome. And what you'll notice is that this red dot corresponds to this green dot. And where it's really red here, it's really green there. And where it's really red here, it's really green there. In other words, that polyglutamine is sticky and sticks the proteasome in one place so it can't do its job. These little bright bodies are what we call inclusion bodies. That's just a fancy term you need to know. When you see inclusion body, you should just simply think it's a protein aggregate. That's it. Nothing more than a big ball of aggregated protein. Alrighty, well, if we're having problems making and folding proteins, that can include the anti-apoptotic BCL2 proteins. So we don't make enough of them, or they're not the right shape, so they don't do their job. That leads to apoptosis. And if we're not properly folding our proteins, the cell will detect this. The unfolded protein response occurs if we have too many hydrophobic residues exposed. That's what the cell is looking for. So hydrophobic amino acids should be hidden. They should typically be in the core of a protein because the protein's surrounded by water. So if the cell finds that proteins are exposing an awful lot of hydrophobic amino acids because, well, either there's mutations in chaperones, so we're not folding up properly, or we have some pathological protein present, the cell then triggers the unfolded protein response, which is it increases the expression of pro-apoptotic BCL2 proteins. And obviously, that'll kill you. The other consequence of a protein aggregation disorder is that damage to the proteosome allows the accumulation of damaged mitochondria. And so we're back to where we were a couple of slides ago. Now we're in the mitochondrial dysfunction. So what they've done over here, C6 is not a spinal level. This is the condition where they've impaired the proteasome. So we have control and then impaired proteasome over here on the right. On the y-axis, we're looking at the amount of free radicals, reactive oxygen species. So as we go up, there's more free radicals. So here's your control amount, impair the proteasome. Notice we have more free radicals floating around. The asterisk is here to tell us that we did indeed have a p-value that's significant, whatever. We have, a 30% increase in free radicals. Those are still going to go about and damage the cell willy-nilly. So they'll donate their electrons, damaging the lipids, proteins, DNA, etc., and eventually leading to apoptosis. So you might have noticed that a lot of these different mechanisms are connected, and you probably have something like this floating around in your head. And as you can see, it's all pretty simple. This is, of course, a joke, right? Of course, I made all these crisscross lines to show you that it is a little bit complicated, but it ultimately comes down to two ways of dying: controlled apoptosis, uncontrolled necrosis. How you get there, there's a lot of ways of doing it. You know, how does ischemia kill us? Well, we don't make enough ATP, and then we, don't clear our glutamate in the astrocytes, so then we get an excess of glutamate, and then that can lead to necrosis. Or it could depolarize us, and again, depolarization leads to excess glutamate release, which depolarizes us further, so this is bad. We got a positive feedback loop here. That never ends well. When we're depolarized, excessively, then we're spitting out too many neurotrophins, so now neurotrophic support doesn't work anymore, and that'll kill you. But of course, we can impair neurotrophic support, not just by releasing too many, but maybe we don't move them around properly because of some protein aggregation disorder. Again, the protein aggregation disorder might not affect axonal transport. Maybe it just screws up the mitochondria, which then will kill you either by leaking out cytochrome C or making a bunch of free radicals that damage the cell and then kills you. Of course, those free radicals could have come from another place, right? They can be produced in the inflammatory response. Remember, reactive glia, like reactive microglia, spit out free radicals. So that's a problem. Hey, inflammation also impairs glutamate reuptake. And oh my God, we've just touched every box. Yes, they are all connected. But that doesn't mean that every single mechanism is always going on in every disorder. So if you've got these big 5 ideas right now, your life will be easier throughout the class because we'll be hitting one or a couple of them for each disorder, and you'll already know how they work. To ensure that you know how they work, make sure you can answer these questions. If you can't, if anything's confusing, put it in the questions box so I know what to talk about in class. I'll see you there.